This unit will cover the cells, tissues, and organs of the gastrointestinal tract. These are the session objectives which you should address when studying this material. Let's start with a couple of definitions. The alimentary canal includes the complete digestive tract, or tube, all the way from the oral cavity to the anus. The digestive system is that entire alimentary canal plus the digestive glands, such as salivary, liver, pancreas, and gallbladder, which all add secretions that are functioning within that system. The gastrointestinal tract includes the stomach, the small intestines and large intestines, and generally the esophagus. This unit will cover all of the organs and regions of the alimentary canal except the oral cavity. The digestive glands will be covered in the next unit and separate from the GI tract. So what are the general features of the GI tract? It is a tube with 4 distinct layers in most regions. It is the major interface between you and your environment. Therefore, it has abundant gut-associated lymphatic tissue for immune response to those antigens which are encountered within that system. It has an extensive vasculature, both blood and lymphatic, and it has its own self-contained enteric nervous system. And I'll talk about that a little bit later in more detail. The gut tube is comprised of four layers, as I just mentioned. The mucosa, which is the epithelium, lamina propria, and muscularis mucosa, an underlying submucosa of dense irregular connective tissue, a muscularis externa, generally 2 layers, but in some regions 3, and an outer connective tissue covering, which is either a serosa or an adventitia. Here's a diagrammatic representation of those 4 layers, the innermost being the mucosal layer, then underlying that the submucosa, and then the double layer of the muscularis externa, and in this example, the outer connective tissue layer is only serosa. So beginning with the mucosal layer, the first portion of that is the epithelial layer, and throughout the entire tube you have only 2 types of epithelial lining, either stratified squamous or simple columnar. There are 2 locations where there are abrupt changes between these 2 types, and that is the first encountered at the place where the esophagus meets the stomach, called the esophageal cardiac junction, because the cardiac is that first region of the stomach, and at the rectal anal junction where you're just about to leave the body. So basically the beginning and the end of the tube have stratified squamous, and the majority of the tube in the middle is simple columnar. So what does the epithelia do? Well, it does what epithelia does everywhere. It provides a barrier between what's in the lumen and what's underlying the lumen, and that keeps out pathogens and antigens and toxins and so on. It keeps the enclosed bolus of food separated from the underlying tissue, and that is maintained via tight junctions. It protects from abrasion at the beginning and the end of the tube using that stratified type of epithelia, stratified squamous. In the majority of the tube, you have areas where you have absorption for nutrients and water. And there's also numerous glandular tissues within the GI tube, which secrete substances such as mucus, which provides lubrication, enzymes for digestion, acid, other buffering substances which help change the pH, as well as enteroendocrine cells which secrete hormones. Underlying the epithelia, you have the connective tissue layer called the lamina propria. Recall that we studied this when we looked at connective tissue in general, quite a while ago now. This is a loose connective tissue layer which contains vasculature, both blood and lymphatic. It contains glands. It contains many different connective tissue cells as well as immune cells. So what cells are in that lamina propria? Well, everything that we previously studied as a connective tissue cell can be found there. Fibroblasts and macrophages, plasma cells, lymphocytes, mast cells, neutrophils, eosinophils, all of those cells that get out of the vasculature, all of the wandering cells, as well as the resident cells that we studied of connective tissue. Other structures found in that layer would be diffuse lymphatic infiltration as well as lymphatic follicles, and those can have germinal centers or not. So you're going to find quite a lot of this gut-associated lymphatic tissue within the GI tube because this is an area where you have all this antigen encountering for the first time. The deepest layer of the mucosa is called the muscularis mucosa. Now this is a double layer of smooth muscle, an inner circular and outer longitudinal bundle. It's fairly thin and it directly underlies the lamina propria. It acts as a separating line, so to speak, between the mucosa and the underlying submucosal layer. Its function is to produce localized movements of the mucosa, which can do a couple of different things. They can dislodge food particles that are stuck or adhered to that mucosa. If you're in an area where you have villi, it can act to separate the villi. It can act to maneuver the glands a little bit. This can all really work towards absorption and secretion functions in a large portion of the tube. This movement is not the same as peristalsis. These are very localized, small movements, whereas peristalsis is carried on by the larger muscle layers that underlie the mucosa and submucosa. So the layer underneath the mucosal layer is that submucosa, which is a dense irregular connective tissue layer, primarily comprised of fibroblasts and collagen. It contains the larger vasculature, the larger lymphatics, and the first portion of that enteric nervous system that I mentioned earlier, the submucosal plexus. In some portions of the tube, you may find submucosal glands, and we'll talk about them as we study the different regions of the GI tube. This image shows an example of the submucosa, which is shown by the blue bracket at the top, and the arrow is pointing out a portion of the submucosal plexus where you can see the parasympathetic neurons and this very small nuclei of their glial cells. And yet another example here is showing you the submucosa over here on the left side of the image. You can see the base of the glands of the mucosa, then the muscularis mucosa here. Beginning about here, you can see the submucosa, which goes all the way to this point here, and then on the right side of this, you can see a bit of the underlying muscularis externa. So this submucosa contains large vasculature, pointed out by these arrows, and here's a portion of the submucosal plexus, as well as all of this dense irregular connective tissue that's comprising this area. Underlying the submucosa is the muscularis externa. This is two concentric, thick layers of smooth muscle. In some parts of the GI tract, you may have a third layer, and I'll go through those when we do the regions. The outer longitudinal layer extends along the length of the gut tube, whereas the inner circular layer spirals around the gut tube. So therefore, it's often called the circumferential. This is the muscle layer that controls peristalsis. Contraction of the inner layer constricts the lumen, compresses and mixes the contents, whereas contraction of the outer layer can shorten the tube. And when both of these layers contract in a slow wave-like fashion, it propels the contents of the GI tract, the bolus of food, through the gut tube. Here's an image where the brackets at the top are showing you the inner circular and outer longitudinal layers of the muscularis externa. To the right of those, you can see the submucosa and even a bit of the base of the glands that are in the mucosal layer. Now, within this muscularis externa, in fact, between the two layers of muscle, you find this second portion of the enteric nervous system called the myenteric nerve plexus. You can see this large portion of it here, kind of circled in the upper image, and then a higher magnification here, where you can actually see these very purplish looking cells with large nuclei. Remember, neurons have a lot of that RER in their cytoplasm, which makes them kind of dark purple. That's called the Nissl substance. And then the small nuclei are the glia that are supportive cells for these neurons. And these are the parasympathetic neurons. The outermost layer of the GI tube is called the serosa or adventitia. This is a connective tissue layer that covers the gut tube. So what are the differences between these? The serosa is a smooth membrane comprised of a thin layer of simple squamous epithelia, specifically called the mesothelium here, and it has a small amount of underlying connective tissue. So the mesothelial cells produce a serous fluid that lubricates the surface of the gut tube, and that reduces friction so that the tube or the organ that it's covering does not stick to other things. Okay, so serosal portions of the GI tube that are suspended in the peritoneal cavity are intraperitoneal. Here's an example of serosa. There you can see that layer of mesothelial cells with some underlying connective tissue, and here's another example from a different portion of the GI tube at high mag.